Welcome to BOC Sciences. Today, we talk about human immunodeficiency virus. HIV is a virus spread through certain body fluids that attacks the body's immune system, specifically the CD4 cells, often called T cells. Over time, HIV can destroy so many of these cells that the body can't fight off infections and disease. These special cells help the immune system fight off infections. Untreated, HIV reduces the number of CD4 cells in the body. This damage to the immune system makes it harder and harder for the body to fight off infections and some other diseases. Two types of HIV have been characterized, HIV-1 and HIV-2. HIV-1 is a retrovirus that may produce an acute, usually self-limited mononucleosis-like syndrome but more inevitably causes a broad array of progressive clinical manifestations, widely known as the acquired immunodeficiency syndrome. HIV-2 causes a similar but far less widespread infection and a generally more indolent chronic disease. The four major routes of transmission are unprotected sexual intercourse, contaminated needles, vertical transmission or mother-to-child through breast milk. Additionally, viral infection may occur through the transfer of blood, semen, vaginal fluid or pre-ejaculate. The 7 stages of the HIV life cycle are binding, fusion, reverse transcription, integration, replication, assembly, and budding. Binding: HIV binds to receptors on the surface of a CD4 cell. This stage can use CCR5 antagonists and post-attachment inhibitors. Fusion: the HIV envelope and the CD4 cell membrane fuse, which allows HIV to enter the CD4 cell. This stage can use fusion inhibitors. Reverse transcription: inside the CD4 cell, HIV releases and uses reverse transcriptase to convert its genetic material HIV RNA into HIV DNA. The conversion of HIV RNA to HIV DNA allows HIV to enter the CD4 cell nucleus and combine with the cell's genetic material cell DNA. This stage can use non-nucleoside reverse transcriptase inhibitors and nucleoside reverse transcriptase inhibitors. Integration: inside the CD4 cell nucleus, HIV releases integrase. HIV uses integrase to insert its viral DNA into the DNA of the CD4 cell. This stage can use integrase inhibitors. Replication: once integrated into the CD4 cell DNA, HIV begins to use the machinery of the CD4 cell to make long chains of HIV protein. The protein chains are the building blocks for more HIV. Assembly: new HIV proteins and HIV RNA move to the surface of the cell and assemble into immature HIV. Budding: newly formed immature HIV pushes itself out of the host CD4 cell. The new HIV releases protease. Protease acts to break up the long protein chains that form the immature virus. The smaller HIV proteins combine to form mature HIV. This stage can use protease inhibitors. Non-nucleoside reverse transcriptase inhibitors inhibit reverse transcriptase by binding to an allosteric site of the enzyme, such as rilpivirine, nevirapine, efavirenz, dapivirine, etravirine, delaviridine, larsuvirine, etravirine and so on. Nucleoside reverse transcriptase inhibitors inhibit reverse transcription. For example, abacavir, emtricitabine, tenofovir, zidovudine and zalcitabine. Protease inhibitors prevent new HIV from becoming a mature virus that can infect other CD4 cells, including aprenavir, darunavir, fosamenprenavir, indinavir and lopinavir. Fusion inhibitors prevents HIV from entering the CD4 cell. Integrase stand transfer inhibitors inhibit the viral enzyme integrase, which insert its viral DNA into the DNA of the host CD4 cell, such as elvidagravir, cabitgravir, tobacistat, dolutegravir, and so on. CCR5 antagonists block the CCR5 coreceptor on the surface of certain immune cells. Senecreviroc is this kind of medicine.